Hello. How are you guys doing today? I am back. Hopefully this is up and going. So I'm going to check everything, make certain we got everything set. I'm going to reconnect whatever I got to connect. Make certain we've got everything. All right. Make certain we've got all that taken care of. I'm pretty certain we are reconnecting on the chat. Hello. Hello. I'm so glad you're here. I'm double checking to make certain my Streamlabs is running because this is the first time. There we go. Okay, yay. How nice is this? I actually have my chat on my Streamlabs now. Hey guys, what's up? So I know I'm on YouTube. Let me make certain I'm on Periscope because it's just me and silly me. You know, that's what we do sometimes. So let me make certain I'm on Periscope. I think I am. Where is... Am I showing up at all on Periscope, guys? Hey, Andy. Hello. I'm going to be really mad if Periscope isn't going live right now, so... Because I'm doing a new thing. It's not showing up on Periscope. What a pain in the butt. Streamlabs OBS, I believe, is what I'm doing. But I'm like streaming to like freaking, this is a pain. It should be up. Let me pull up one thing. You know, I can't just have regular stuff ever. It's just like really hard. Restream. I need to find out why Periscope isn't working right now. It's sending data. It's showing it's sending data to Periscope. Showing that it's sending data. I'm on YouTube. Maybe Periscope is just buggy today. I don't quite know what's going on. Weird. All right. Yeah, I'm trying Streamlab because, I mean, it's supposed to be seamless, so I'm giving this a go and see what happens. But it seems like it's being a bit of a butt for my Periscope chat. Doesn't seem to be going on right now. Let me see. Go to broadcasts. Yeah. Well, that is just a booger. I guess I will have to upload this later on Periscope. What a pain in the butt. Oh well. So we've got, it's still connecting. It looks like it's still connecting. I can't just have regular stuff happen, you know. It's just always got to be something. It's always got to be something. Oh well. Well, let me give this a go. I'm already a bit late, so I don't want to be later than I already am, you know? So, yeah. Okay. Fun times, huh? So, today we are going to talk about bacteriophages. Boy, I'm upset that it's not on Periscope. Maybe it's just taking a while or something. I don't know. So, all right. So, welcome to the show. This is the first time I'm doing Streamlabs, and so I might actually have to go back and redo this on Periscope, or at least upload a video there and give that a go, because it seems like it's not connecting to Periscope. And so I can never just have any stream without some kind of technical difficulties. So this is the same it was just kind of streamless from OBS to Streamlabs. If you are on Streamlabs, you can send me like a tip through Sunday Science is making you tardy. Yeah, probably. If you want to send a tip or something, if you like what I do, you can go on streamlabs.com/ScientistMel. That's where I am. And you can kind of hit me up there if you want. So I have like one alert. I don't know what this is. Click to read your unread notifications. Fantastic. This is a new thing on Streamlabs OBS. It's just skip frames detected. Okay, thank you, Streamlabs. I don't know how that's useful to me right now. I'm still not on Periscope yet. What a pain in the butt. I don't care for this. Okay. Anyway, maybe I'll just redo this tomorrow on Periscope with OBS and see how that goes. Okay, so today we are talking about bacteriophages. That was one of the things that won last time. So it was really, really close. I had like a three-way tie on, almost a three-way tie between beer and barbecue, and bacteriophages and photons. And so now we're talking about bacteriophages. So let's go ahead and get started. Else note where you can't see it, so that's always nice. So I'll have to fix that for next time, but at least you heard the pretty catchy music, so that's kind of cool. So bacteriophages, woohoo. There we go. All the clicking. All right, working the format, man, it's pretty weird. So what are they? What are the types? What do they do, and how are they useful to us? Because, there are some bacteriophages that are actually indirectly lethal to humans. But the thing is, bacteriophages only infect bacteria. There are viruses that go and they attach to a specific bacteria, and they inject their genetic code. Now, they contain either DNA and RNA. They can have anywhere from 4 genes to hundreds of genes. They're considered the most abundant organism. I use that term loosely because they're not really considered to be alive. They're not really living. Viruses cannot reproduce on their own. They have to hijack cells, in this case bacteria cells, in order to make their parts and make more copies of them. It is thought that bacteriophages are a huge driver in bacterial evolution. So what makes up a bacteriophage? Let me make certain you can see that. Oh, look at that. I am like covering some of this, so I'm going to just go to the big monitor so you can see there. All right, so here we've got the different types of bacteriophages. The capsid of a bacteriophage holds the actual genetic code. Now, the shape of the capsid can vary. It can be what we call icosahedral, it can be filamentous, or it can be what we call a standard head-tail. Now, the head-tail structure seems to be exclusive to just phages as well as their relatives. It is not found in eukaryotic cells. So, back to this. Bacteriophages are viruses that infect bacteria. They come flying in through the extracellular space attached to the bacteria, and then slowly inject their code right into it. So it's pretty cool on how they do that, but it's still kind of scary nonetheless. Bye, Brian. Now, there are the life cycles of bacteriophages can be what we call lytic and lysogenic. This is actually pretty important because the two are very, 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 very different. So what's the difference between the two? There we are. Pop-up chat on the side. How you doing? How you doing? Bacteriophages, essentially. Bye, Brian. This is what they do. They have two specific types of life cycles. They have the lysogenic and they have the lytic cycle. Now, Khan Academy, I don't know if you're familiar with them. If you're not, then you probably, I don't know, you haven't really gotten involved in science education or math education. It's a fantastic website. So if you're a teacher, if you're somebody who's trying to learn a particular topic, check out that site. I like the way that they've broken down the lytic cycle as well as the lysogenic phase. So I'm going to kind of pull a bit of their resources for this. So I'm going to give credit where credit is due. So there's specific stages of the lytic cycle. Now we saw a little bit of that picture before, that GIF image where we saw the bacteriophage, the head tail was landed on top of the bacterial cell. Well, proteins are in the tail of the phage, and they come down and they bind to a very specific receptor. These types of receptors are usually made of different types of sugars attached to maybe a glycoprotein. The protein is embedded in the cellular membrane, and then you have this little antenna that kind of sticks outside of a bacteriophage, or outside the bacteria for the phage to attach to. It's highly specific. And so once these attachment proteins come out from the tail of the particular bacteriophage, the entry phase begins. And the phage injects what we could have like double-stranded DNA into the cytoplasm of the bacteria. Now, DNA copying then happens along with protein synthesis, and that phage DNA is actually copied, and the genes are expressed to make the proteins, such as the capsid proteins. So the bacteriophages essentially hijack the resources within the bacterial cell, and it goes through, and the ribosomes will read the particular genetic code, and they'll make the proteins associated with the bacteriophage, associated with generating parts of the bacteriophage. Hello. Yes, I took a break for 2 weeks so I could settle into the new job, but now I'm back with the cool stuff that is bacteriophages. So I'm super glad you're here. So what happens then in the lytic cycle, all of these bacteriophage parts are made within the bacteria. And so the capsid assemble from the capsid proteins, and then those capsids, like the heads of a head tail makeup, are stuffed with genetic code, and they're also put together with new phage particles. So later on in the lytic cycle, the phage starts to express genes for proteins, and it starts to poke holes in the plasma membrane. That's the outer portion of the bacterial cell, and some bacterial cells have cell walls like plants, and so they'll start to poke holes. It's actually kind of gory and violent. They'll just and they'll go through and they'll start poking holes in it, which is actually kind of scary. So then what happens is those holes, we have a compromised membrane, start to allow water to come rushing in, and we have explosion of that once, happy bacteria cell. Now, all of these new phages are then released out of the cell, and that allows the process to continue on some other neighboring bacterial cells. So, this particular process with the lytic cycle is gory and violent, as it seems to be. What it can do is these hundreds of new phages can then find and infect all of the other bacterial cells nearby. And in this way, you don't need a whole lot of lytic cycle to happen because one infection can let phages spread kind of like a wildfire throughout a bacterial population. So, in essence, if you have one of these types of bacteriophages that attaches to a particular species of bacteria, it can release hundreds of phages in one lytic cycle, thereby spreading the infection super freaking fast. Hello. No, don't worry about it. For whatever reason, Periscope isn't working right now, but Twitch is, so it's strange. And yes, plug for the Non Sequitur show. I am appearing on that. Is it 9/11? Is that... no, it shouldn't be in 3 days. I thought I had more time than that. I think, isn't it the 14th that I'm appearing on the Non Sequitur show, talking with Jilly Juice about stuff she thinks you can drink something and grow new arms. So that'll be interesting. Little plug on the side for that. Hello. So back to bacteriophages, since I had a hey squirrel moment and somebody said something about a show I'm going to appear on later. Woohoo. So the lytic cycle is pretty gross for bacteriophages. So let me show you a bit of what we got here. Okay. So this is kind of like a graphic. And if you have a graphic, in this regard, we've got the various different types. I guess I'll just, because I'm covering up some of it. Here we go. We got the various different types of the portions or the stages of the lytic cycle. We have attachment of the phage to the bacteria. There it is, and it just plugs right in. And it's just like, hey, guess what? I'm going to start entering all of my phage DNA into your world, and then I'm going to wreak havoc. And so all of a sudden we got the phage DNA that's being copied, and all these little capsid proteins are now going to start assembling themselves, and then they start poking holes in the membrane, and then it releases it into the extracellular space to go and attack a new bacterium kind of hanging around. So it's actually pretty gross. You know what I mean? It's actually pretty gross and mean, you know. I'm just like, gosh, you know, just the lytic cycle is not fun for a bacteriophage, or at least for the bacterium. Bacteriophage don't care. All right, so the lysogenic cycle. The lysogenic cycle is the other version of how bacteriophages reproduce and what their life cycles are like. So this particular cycle allows a phage to reproduce without killing its host. Now, some phages can only use the lytic cycle due to the nature of what they are. But the phage we are following, in this case, we can switch between the two cycles. Now, in the lysogenic cycle, the first two steps where you have the attachment and the DNA injection, those are exactly the same in the lytic cycle. But once the phage DNA enters inside the bacteria cell, it's not immediately copied, and it's not immediately read, and proteins aren't made right away. Instead, it recombines with a particular region of the bacterial chromosome. This causes the phage DNA to actually become a permanent part of the bacterial genetic code. So, I'm going to show you the lysogenic cycle. I know you guys are excited. All right, so here's the lysogenic cycle. So first and second phase is the same. It attaches to the outside of the bacterium. It releases the phage DNA. It's just like, hi, how you doing? I'm attaching to you. And now I'm going to put all my junk in there. And so then what happens is it becomes a part of the bacterial DNA. And it's what we call a prophage. Not prophase, that's something else. Prophage. And so it's continued. And so what happens is you have cell division. So whenever you have a cell division through binary fission, that's the type of cell division bacteria undergo. You have a copy of the prophage genetic code that's incorporated permanently into the bacterial cell. And so doesn't kill the cell. What it does is it makes the cell undergo, it actually becomes a permanent part of the cell and then transforms that bacteria, which is kind of cool. So, boom. I'm trying to do the interface of this software and it's just kind of weird going back and forth. All right, so let's get in a little more detail about the lysogenic phase. Most phages are either lytic, purely lytic, sometimes those are called virulent phages, or they are capable of switching between lytic and lysogenic cycles. And so sometimes those are called what we call temperate phages. Now, when it comes to virology, there's almost always an exception to just about every single rule. Now, this is true for the phage life cycles. Now, you noticed it before when we were talking about the different types of bacteriophages. We had the capsid, and then you have the kind with the head tail, and then you had that long little line, which is filamentous. That's that long rod-shaped type of phage. Those are secreted from the cell in a process that does not lyse or kill the cell. So you can have that particular type of phage that actually managed to wiggle its way out of the bacteria cell without causing it to burst or be all like graphic like that. So, but even though the cell is actively producing these new phage particles, this is like a lytic-like life cycle, but it doesn't actually involve the cell to be destroyed or lysed or burst or any of that, wow, kind of weirdness. Now, not all phages integrate their DNA into the genome of the bacteria, their host cell, during the lysogenic cycle. So it doesn't always happen that way. Some kind of keep their genome in the cell as a separate circular piece of DNA. So that particular DNA can float around in the cytosol, because the thing is bacteria do not have a nucleus. It's just, the code is floating around. It makes it easy to manipulate. Now, even though it's still floating around in the DNA and not in the cytosol and not a part of the actual genetic code of the bacteria, this is still considered what we call a lysogenic cycle, because the phage does not drive production of new virus particles that cause the death of the bacterium cell. Instead, it kind of remains kind of silent, hiding out, and is passively copied along with the cell's DNA as it splits, which is kind of cool, right? So bacteriophages are pretty neat in that regard. All right, click. I know y'all can hear my mouse clicks. All right, so here's the other thing that we got to kind of think about. How does a phage decide or choose, essentially, whether to enter the lytic or the lysogenic cycle? The lysogenic cycle is that sleeping cycle, remember, where it just kind of hides out and becomes part of the bacterial genetic code. The lytic cycle is the one where it bursts. All right, so how does it decide which way to go? So one important factor is kind of think about, if you have a lot of phages infecting the cell at once, that can cause co-infecting phages to make it more likely that the infection will use the lysogenic cycle. So instead of just if it's like a heavily infected bacteria, it might just do that sleeper cycle instead of the lytic cycle. This kind of helps prevent the phages from wiping out the entire population of bacteria, because you don't necessarily want to kill all of the bacteria all at once. So if you have a heavy infection going on, it would make more sense for it to kind of enter that lysogenic phase, so that would allow the virus to kind of stick around a lot longer. If you end up just destroying all the bacteria, then there's nothing there for it to infect. So this strategy kind of helps prevent that wiping out. So if the phage to bacteria host ratio is too high, the lysogenic phase tends to be, the lysogenic cycle tends to be much more, tends to be selected for natural selection, right? When there are multiple phages infecting a single bacteria, This is suggesting, like statistically speaking, that the phages would be in danger of wiping out the host. So lysogenic in that case. Now, phages that flip to the lysogenic mode in response to high infection may be less likely to run out of host. Now, what triggers a prophage to pop back out of the chromosome and enter the lytic cycle? That's where it's the gory, we poke holes in the thing and blow everything up. But at least in a laboratory, DNA damaging agents like UV radiation and chemicals, essentially carcinogenic type of things, will trigger most prophages in a population to reactivate. So that can kind of wake it up. However, a small fraction of these particular bacteriophages, or the prophages, that's the genetic code that's within the DNA of the bacteria in a population, kind of spontaneously go lytic, even without these external cues. So you may not have an external cue that causes this to just go, oh, wait, hold on, let's bust this cell open. But there is something, there are some studies, as I mentioned, in a lab where they've been able to introduce UV radiation, other types of carcinogenic agents, in order to get the lytic phase to turn on like that, which is really kind of cool. All right. So what about the bad ones that hurt people? Technically speaking, there aren't any. There we go. Did I get it right? There, I got it right. Man, this one's kind of weird. I don't know if I like the Streamlabs so much. It's kind of it's kind of being a bit weird on me. So when we're looking at bacteriophages, we do have bacteriophages that cause bacteria to become extremely lethal and harmful to people. So some bacteriophages induce toxin production in bacteria that leads to human illness. Now, the bacteria in itself isn't harmful, but becomes harmful when infected by a bacteriophage. So we have specific bacteriophages that infect certain types of bacteria. We have cholera, diphtheria, hemorrhaging diarrhea, botulism, and scarlet fever are all caused by bacteriophages manipulating bacteria to produce terrible toxins that make us incredibly ill. So that kind of sucks. But that's one way that bacteriophages actually do more harm than good. All right, so popping back over to here. Excuse me. I have been talking a lot. Now, since the genetic code is universal, these bacteria-infecting viruses utilize the ribosomes, an easily modified genetic code of bacteria, no nucleus to kind of house it, and they alter the bacteria to produce these toxins. The bacteria reads the code injected by the virus and follows the directions, generating proteins that make us crazy ill, and oftentimes it is lethal. There's something interesting about the fact that these viruses can genetically modify bacteria. Is this a potential solution for the antibiotic-resistant bacteria? So let's have a look at the history of using bacteriophages to combat harmful bacteria. All right, what's that about? Here we go. There's some controversy about when bacteriophages were discovered. It was possibly first discovered in 1896. I'm sorry, that should say 1898. My 8 dropped off on the end there, but hopefully you guys can, extrapolate from less information, Those are the two types of people in the world, those who can extrapolate from incomplete data, and I like that joke. But it was technically first discovered in 1915, maybe, but it was it was primarily discovered to be lethal to bacteria in 1917. There's a lot of controversy surrounding that because, science is never just an easy thing, and people are always fighting over who did what and when, as you do in science. So Ernest Haenken, let's start in 1896. Ernest Haenken was a bacterial biologist, and in 1896 he stated essentially that antibacterial activity against Vibrio cholerae was observed in the Ganges and Jumna rivers in India. It was his thought that this unidentified substance was responsible for this antibacterial activity, as well as limiting the spread of cholera epidemics. They didn't know at the time that the cholera bacteria was being infected by this bacteriophage. They just knew that this bacteria was around causing people to be sick. Well, 2 years later, there was a Russian bacteriologist, Gamaleya, observed a similar phenomenon when he was working with Bacillus subtilis. That's a really nice rhyming species, isn't it? It just makes you want to say Bacillus subtilis. That almost sounds like a Harry Potter spell, doesn't it? Bacillus subtilis. It was completely unrelated to what we're talking about, but that just made me think of, that was kind of fun to say, and also could be a Harry Potter spell. It wasn't until Frederick Twort, who is an English medically trained bacteriologist, who observed the same phenomenon that these other two investigators saw. But he was the one that was first to suggest that it could be a virus. Now, the fundamental textbook discovery of these bacteriophages that were later found to be lethal is credited in 1917 to Felix de Herelle, a microbiologist who was at the Pasteur Institute in Paris. So, who discovered bacteriophages? I don't know. Everybody fights over stuff like this. It was this person. No, it was this person. No, it was this person. Who did it? Who knows? You like the Vidians from Star Trek Voyager better. So, antibiotic resistance among the pathogenic bacteria is becoming a serious issue as infections are harder to treat. The use of bacteriophages to kill specific bacterial pathogens is gaining more ground as a potential treatment for infections. Now, bacteriophage therapy was widely used in the 1930s and 1940s. It was also used with de Herelle in the Pasteur Institute. Once he found that it was lethal, he started to use it to cure various forms of diarrhea. Now, this particular type of therapy is still used in portions of Eastern Europe and the former Soviet Union. Now, in the West, a lot of the research and use of bacteriophages to treat bacterial pathogenic infections was kind of scrapped and completely abandoned once antibiotics hit the scene. Now medical science in the West is moving to use more bacteriophages in treatment of these infections, as these particular bacteriophages do not harm human cells. Now, with the emergence of immunotherapies for treatments of more debilitating illness like cancers, incorporating this well-established research of bacteriophages into medical treatment of bacterial infections, this is an emerging answer to the rampant issue of antibiotic-resistant bacteria. So why not get bacteriophages to go and attack it's a highly specific interaction, so it doesn't harm human cells, because we don't have the receptors, the same receptors as bacteria do when it comes to these bacteriophage type of infections. So that's where medical science is moving towards, to kind of boost and promote new ways of treating bacterial infections, especially when you look at staph infections that are really, really horrible, especially in hospitals. Perhaps they can help work towards making a bacteriophage that will exclusively attack that particular staphylococcus infections. So this is kind of where medical science is moving towards. So that has been, what have we talked about today? Wrapping up everything. Bacteriophages. They're not necessarily considered living, but they have been a driving force in bacterial evolution in the fact that they're able to completely modify the genomes of different types of bacteria. They contain DNA or RNA, anywhere from 4 genes to hundreds of genes. They're considered the most abundant organism, but they're not really living because they cannot reproduce on their own. So this makes them inherently useful, especially when we consider their uses in medical science. So, what have we talked about? What are they? What are the types? What do they do? How are they useful to us? So we've kind of covered what bacteriophages are, what the types, what they do, how they're going to be used in medical science. This inherently gives them a bit of an edge. So I want to take a moment to show you all of my sources. Hold on, you can't read that. Fear not. I'm going to put these slides on my website. You can download all that. There's so much information available on bacteriophages and how they're used, the history, how they've been found. Just everything. There's a little nod to Khan Academy. Go check out their site. It's pretty awesome. And let me thank all of my patrons. So here we go. Thank you, my super awesome patrons. They help me keep the lights on. I've branched into promoting my work through social media. So pretty much all of the funds that I get go towards paying for my website. I've been able to buy new tech. I'll be using green screen soon once I sort out the better lighting with that. So thank you, Tony, James, Lauren, Jen, Carl, Melanie, Patrick, Daniel, Stephen, Paula, Tim, Carrie, Cersei, Keith, Duke, James, Andy, Zachary, Tony, Bo, Stephen, Sarah, Chris, Graham, Dragnot, Godless Iowan, Jennifer, Corey, and Heavy. Thank you for what you thank you for what you do. You help me keep the lights on, and I'm now editing new podcast episodes with my new co-host Ashley. Do check her out. She's known as Bio Mermaid on Twitter. Yay! Bo and Andy are in my list. That's awesome. Where can you find me? Everywhere on the internet. ScientistMel.com, Patreon, Periscope, YouTube, Facebook. I'm on Twitch, and I have a Discord. And unfortunately, this one didn't air on Periscope, so I've got to sort that out and hopefully do that later. Go check out my Facebook page and join in the talk for that. And that has been the science of bacteriophages with the wrong date on my image there. That's not August 18th. That's when I did beer and barbecue. You see, I'm always just kind of silly and not able to like properly fix, I don't know, my chat. So if you got questions, I can chat for a bit. We got like 15 minutes left before I pop off of here. I started a couple of minutes late, and I still managed to like not get on Periscope, which is weird. So yeah, you can use Hey ScientistMel on Twitter for questions. I highly recommend using that hashtag. I spent about 5 minutes this morning trying to go through all my mentions on Twitter. I was just like, okay, I'm in like 4 conversations I didn't know I was in. So let me go through and get all of this done. So it's pretty weird. It's great. I like Twitter because I'm able to get that, get that there. I'm also on Curious Cat. So if you're on Curious Cat, you can send me a question there directly, and that's another way for me to find your questions and answer them, and then I can post them on social media. So I think it's curiouscat.me/ScientistMel. So you can ask me a question there. I hope you guys have enjoyed bacteriophages. This has been pretty neat and pretty groovy. I like being able to talk about kind of cool, weird stuff that looks like it's from outer space, doesn't it? Bacteriophages are weird. I wonder how well they would do and just because we know what I want to find out is, can bacteriophages withstand like the vacuum of space as well as cosmic radiation? Because we know tardigrades can, but can bacteriophages? That would be pretty interesting. Wonder if I can get an experiment sent up on the next ISS mission. I know a teacher who does that. Let me contact them and see how it is that they got their stuff on the net, on like the ATK orbital. Because I met a teacher that actually had their stuff go up on the ATK orbital mission to the ISS back. I think that was in May. Was that in May when I went? I think that was back in May when I saw that particular launch. So I should communicate with him and be like, hey, what's up? How'd you get your stuff up there? Oh my goodness. So, do you guys have any questions? None at all? CuriousCat.me. Thanks for the link, Bo. You can ask me questions there. I've gotten lots of interesting questions. You know, for whatever reason, people seem to think it's 12 years old. Yay! It was. Yay. I'm so glad you guys enjoyed that. Oh my goodness. I'm sitting here drinking my coffee. It's almost 11. I don't think you guys know how much work I put into this. I spent most of my morning trying to set up Streamlabs, and then I knew enough about bacteriophages. I knew enough about bacteriophages to where it wasn't so, like, you know, heavy on trying to put together a talk for you guys. May 20th was the launch date. Thank you, Bo. Bo is just like my guy who knows like random stuff and helps me out and stuff. It's pretty cool. Do tardigrades get their foreign DNA from bacteria or from contamination? Huh. You know, I wonder that. That's a good question. May 20th. Okay, so I don't know. I wonder. I would think that they would be able to interact with a lot of things, possibly things that we haven't fully discovered yet at the tardigrade. Okay, any bacteriophages that might take out cancer cells? Huh. That's a good question. Caffeine addict. I don't drink coffee past noon because if I do, then I will be up till 3 in the morning. Caffeine does that to me. All right, Andy's question. Any bacteriophages that might take out cancer cells? That's an interesting question. The only thing that I think there would be an issue with that is if there are any cellular receptors that happen to be on non-cancerous cells as well as cancer cells. You do have, like for instance, cancer cells, they're able to trick the immune system. So they might have certain receptors that allow them to communicate with the immune system. So if they still have like normal cell receptors, if that happens to be what the virus attacks, then you could have a possibility of it attacking human cells. But then they wouldn't be considered bacteriophages anymore. Bacteriophages are exclusive to bacteria because they have a different set of receptors on the outside that they attack. So they would never attack human cells. So they would just be like regular viruses. But there is research that has been performed where they have taken viruses that infect the immune system, an example being HIV, and they've reprogrammed it where they've taken out the code that causes human illness and put in code that rewires T cells, the immune system, to specifically attack a person's cancer. So that's a new thing, and it's gaining ground and for it to work. And so now we have potential immunotherapies where people have some of their T cells removed from the body. Then they inject like the viruses to go and attach to the T cells and completely rewire that T cell's genetic code to where it's meant to go specifically attack the cancer cells. That has actually happened. If you haven't checked out, look up on the internet, on YouTube, stuff and things. Go look up on YouTube the HIV used to kill cancer. That video doesn't make you cry. I don't know what will. They reprogrammed a little girl's immune system to attack her cancer. She was on her deathbed. This was a last-ditch resort to save her life because the chemotherapy didn't work, the radiation didn't work, nothing worked. And so they ended up reprogramming her immune system to attack her cancer. This was it almost killed her. It almost killed her. Like within a few days, she had like really high fever. Once her immune cells were injected back into her body, it went through and immediately just started taken out her cancer. It's amazing. And she's been cancer-free since. I won't cry now. Oh my goodness. Oh, it's just so, you know, you're just like, you know, you get a little, every single time, whenever I taught biology, I would always play that video for that. I would just feel like, oh my goodness. Ah, it is very awesome. Go check out that video. It's great. It's GE Life Sciences, I believe, funded that particular study, and it was at Johns Hopkins. Why am I telling you that story? Because it is a hopeful story. Oh my goodness, it's a hopeful story. But yeah, so to treat bacterial infections, they're moving back towards bacteriophages because they're highly specific and only attack the bacteria. So, modifying, essentially going through and working through that and kind of just, you know, attack the bacteria. So it's neat. It's neat. Why am I telling you that story? It's a beautiful story. It's not like a sad ending. It's a happy ending. Oh my goodness. I have got to work out the technical kinks with this. So if you want to do like a tip during the show, you can go to streamlabs.com/ScientistMel. I think a zombie pops up or something and goes rawr, and you can send a message or something. I think that's how you can do like super chat and how I pour bacteria. It's not poor bacteria if they make you sick. It's poor bacteria if it's you know, what if it's a harmful bacteria? You know, like the cholera inducing one. Yeah, but the cholera inducing one, that's already been modified by bacteriophage. Did that work for meningitis? Maybe. That might could work for meningitis. There's different types of, is it certain types of meningitis that can be like lethal within a few days, especially among adolescents? Is that it? Which one is that? Now I have to go and find this. Meningitis. Is it that one? Ah, there you go. Thank you, Bo. Emma saved by using a modified version of HIV. If you've not checked out meningitis. Okay, meningitis adults. Well, adolescents. Adolescent meningitis. Yeah, I think so. But that's through a vaccination. So if you have a vaccine for that, isn't vaccines, that's primarily for viruses. I'm not keen. They need to live their lives. Okay, meningococcal conjugate vaccination. Okay. It's been a while since I've looked through on meningitis. I'm reading the CDC site on that. Okay, 4 serogroups. Meningococcal vaccines. It's like, okay, alrighty. This is usually how I read. I read through stuff, and I'm like looking through all this like really quickly. So you can get vaccinated for that. Meningococcal conjugate vaccines are safe. It says it right there, anti-vaxxers. Go away. And you get a booster dose every 8 weeks. Oh, you had bacterial meningitis. Okay. Yeah. You know, you would think it could do that. Gosh. So, I did do a talk on vaccines a while, you know, on my website a while ago. On the YouTube channel. You can go check that out if you want to learn more about how vaccines work. I need to do more shorts. I need to do more shorts episodes. So maybe I'll do my shorts on bacteriophages. I think I did a shorts on vaccines. My ScientistMel shorts. They're like 15-minute videos, 10 to 15-minute videos. If you don't want to sit through a whole live stream, you can do that. That crap almost killed you in high school. That sucks. Yeah, and you can actually spread it through sharing drinks and stuff. So that's gross. You know, it's like, here. Then you just spread throughout a population. Oh my goodness. I'm trying to get people to stop using hand sanitizer because now that's contributing to antibiotic resistant bacteria. So I'm like, stop using hand sanitizer. You're making everything worse. So the few bacteria that live, they, they're freaking resistant to hand sanitizer now. Like, you're killing us with your freaking hand sanitizer. I've actually had people go like, why can't I use hand sanitizer when I leave it when I leave lab? When I was teaching at the university, the students there would be like, why can't I use hand sanitizer instead of washing my hands? Like, all you're doing is just squirting ethanol on your freaking hands, man. You're just moving the chemicals around. You're not like actually washing them off. Hand sanitizer does not get rid of chemicals that you encounter in a lab. So it's just getting people to understand hand sanitizer is not the same as washing your hands, for whatever reason, should not be as difficult as it is for some reason. People think, oh, I'll just use hand sanitizer right before I eat. I'm like, all you're doing is that's not the same as, there's a reason why you wash your hands, to get all the crap off your hands. You're just moving the dirt around a little bit, You're not actually killing anything. So, I mean, you're killing the germs, but you're also killing beneficial germs that aren't really, harmful to you, but harmful to the bad stuff. You're killing that too. It just makes me nuts. I'm like, why are you using hand sanitizer? There's no reason to do that unless you have a compromised immune system, and anything will kill you. Oh, goodness, that sounds painful, Andy. I'm glad you're okay. That sucks. Nobody wants to go through that. Nobody. There's Einstein still hanging out behind me. I haven't done my show in a while. So, upcoming things that I'm doing. So, I was at Nerd Night. If you have not joined any kind of Nerd Night talk, you should totally do that. They're all over the U.S. Look up Nerd Night in your area and check it out. So, check out Nerd Night in your area. I've done 2 Nerd Night talks. I'm looking at starting a local show, local version of my Spirited Science show, which is long overdue for a new episode. I really need to do a new episode of that. But I found a local venue, so I will be streaming live from a local venue like a pub or a restaurant or a brewery. I'm trying to line up different venues, and I need to get speakers together, and we will be doing Spirited Science live streaming from a bar or a pub or something like that, where we will incorporate science and adult beverages. It's just awesome. So we'll be doing that. Hopefully I'm gonna get a date set up in October and get my speakers together. Nerd Night is awesome. I've done 2 live shows with Nerd Night, and last one was about how to read scientific papers and spot bad science. And so we ripped apart Goop and that nonsense, which, by the way, she's having to pay for a settlement about her jade eggs. Woo! One for the smart people who knew not to use them anyway. But, she's having to settle a lawsuit for claims, what they do, and they don't. They don't do what they claim. So there you go. Oh, I'm so glad I had you on the show, Andy. That was a lot of fun. You were on the Tesla talk, so that was cool. We learned a lot about that. Hey, Steve. Hello. So I'm still trying to get together my Katherine Johnson talk. I've been waiting on a person to get back with me. I may just have to just say, I'm sorry. I've talked to you several times, and you said yes, and I guess she's really busy. So I'll have to get that together. So gonna get Katherine Johnson. I'll have my shows at stream from my house, but I'll also have the live shows. I've got to start getting together speakers for the local show. I could probably go talk to my contact at Nerd Night locally and see if they can push people my way and see if they want to do that. So that'll be cool. And I have found a venue here for the local shows, so that'll be neat. I'm excited about that. And I'm appearing on the Non Sequitur show, I believe, next Friday, where I will be opposite Jilly Juice. If you've not checked out that, go read that. That's weird. She thinks you can drink certain juice and grow new limbs. So I'll be pulling together a bit of research and be talking to her on the Non Sequitur show about that. So I'm curious about how that's gonna go. You never know. I've read some of her stuff, and I needed to take a break because, wow, wow, just like, oh my gosh, drink this juice. And apparently, Kyle from the Non Sequitur show said that she's also made claims that if you drink this juice, you won't be gay anymore. So I wonder how that works. Are all papers on PubMed peer-reviewed? Here's the thing that's interesting about PubMed. If you have to go and look at the journal that it's published in, because even Elsevier has a homeopathy journal. So I'm just like, okay, homeopathy is just not really a thing that we should have, because it's not real. So you can find publications on PubMed in peer-reviewed journals, but you need to go and look at these journals, because that doesn't, that doesn't mean anything. Yeah, I guess, so be cautious of these peer-reviewed journals. Make certain you're looking at the actual journal and you're like, oh, this is homeopathy. Oh goodness. Dr. Phil had Jilly Juice on the show. Find it all on YouTube. Oh my God. So I'll be opposite her. She was on Dr. Phil's show already. That just, credibility. If Dr. Phil had me on his show, I don't know. I think I'd laugh the whole time. Like, what are you a doctor in? First of all. Secondly, it just, man. I wonder, if Jilly Juice's stuff can make you not gay, is it possible it can make you gay? Because that would be interesting. Does it have, does it go back and forth? Is it just a matter of, do I add more carrot to it and it makes me gay or not gay? Or do we add more lemon and all of a sudden, lemon and its super powerful qualities suddenly make you not gay? I mean, is there a thing? What exactly is it? Is it like the addition of a specific herb? Do you add glitter? Maybe the introduction of glitter makes you gay. Is that it? Or if you get, I'm just kind of like, it's just weird. How does something make you not gay by drinking it? I'm suddenly not gay because I had this water that was purified by banging it on a horse or something. It's weird. Oh, Alex Jones. I can't ask him. He's not on Twitter anymore. Some of the funniest things, I don't ever watch any of Alex Jones' stuff, but some of the funniest things I've ever seen is somebody had taken all of his rantings and turned it into a song with a guitar. And it was just like a slow song where he was just kind of upset and sounded kind of sad. It was funny. Oh my goodness. You've just read a paper claiming that DNA doesn't determine the body plan and body form on PubMed. Well, I would have to read the paper to further comment. I can't comment, further on that unless I read the paper. You might be misinterpreting it because you have, it's not just DNA. When you look at the formation, okay, here's a little bit on that. Okay, hormones play a role too, but it's not just DNA. You have women with a Y chromosome. Okay, you have women with a Y chromosome. And so the default for nature is female. Okay, so as a baby forms in the womb, you have certain triggers, hormonal triggers that have to happen. And so the presence of the Y chromosome is supposed to shut off the "don't be a girl" stuff, right? So the don't be a girl stuff shuts off. And then all of a sudden you have the production of different things. And then you have the stopping of the formation of, the ovaries and all that. So what happens is, the reason why there are some women with a Y chromosome is because their cells don't have the receptors for the hormones that turn them into men. So they have testosterone in their body that's, being produced, but since their cells don't have the receptors to accept that particular hormone, they become women. Okay? So the body plan, it's complicated. It's not just exclusively DNA. So there you go. You can also ask me questions on my Discord. So there's that. Let me go back through because I've had a lot of questions come through. Since I haven't been here in a while, I want to make certain I kind of help everybody out. Yay. You can send me the link for the paper on Discord. Jelly juice will get rid of the gay. Yes, I was talking about that. I mentioned that. I wonder if there's a jelly juice that can actually make you gay, because I would like to try that too. I think that might be neat if we're going to do that. Here's the mix. Yeah, check out my website too. So those are bisexual ducks. Somehow I've missed part of a conversation. The paper holds that DNA chemistry can't hold information for the body plan and form. I don't know. I need to read that before I can further comment on it. That's one of the things. I can't just, you don't have Discord? Well, send me the link on Twitter with hashtag HeyScientistMel. You can send it to me there. I'm also on Facebook, facebook.com/ScientistMel. You can also put it in the comments on this particular video. I have so much admin I have to do for the videos now. I have to go fix my thumbnails. I have to go change the descriptions and, and all of that. So, if you can't be a patron on my Patreon, then shoot me a tip. Streamlabs.com/ScientistMel. Woohoo. And you can do that. Discord scares you. It shouldn't. Don't let Discord scare you. For whatever reason, I've tried to turn on the notifications on my app on my phone and it doesn't do it. So there's people that'll send me messages. But fortunately, I have other people that'll send me messages. You got a question on Discord? And I'm like, thanks so much. And then I'll go and check it and then it works out. Oh my goodness. Oh goodness. Bo, thank you for your help on that. So we got it. You'll place your spam with a bot. He's not spamming. It's all right. Bo actively takes a role in helping me out. So I've been on here for an hour, guys. Oh, it's a good idea. Okay. Thank you to the people who are tech savvy. Look a Pig helped set up my Discord, which was nice. So that was cool. Thank you for that. Well, nod to him for that. That helps me out. I will have a look. It's making you so confused. Okay, I'll have a look. Just put it in the comments and I'll have a look. That's cool. Bisexual ducks. It happens. There are ducks who actually are necrophiliacs too, and will have sex with dead ducks. That's a thing that happens as well. Ducks are pretty unusual in their sexual activity. Perhaps we should, and ducks was actually a topic on the poll, so I'll throw that back in and see if we can have the science of ducks and all the weirdness that goes on with them. They have like crazy like corkscrew penises and, fake vaginas. It's ducks are quite unusual, and they have sex with dead ducks. It's weird. It's weird. Post the link. All right. Yes, volunteers are awesome. Thank you. And I'm pretty well settled in my new job. I'm back in academia and working on stuff there, so that's cool. So having to navigate that out of industry and back into academia feels like I'm going home again, so that's nice. So ooh. Yes, ducks Dogs are weird, aren't they? I love weird. Well, I'm going to hop off here. Thank you guys for being a part of the show. I will be back next Saturday. This weekend, I will be editing new podcast episodes for Hey ScientistMel. If you're on Patreon, you get early access to those by a week. And I have a new host named Ashley. She's fantastic. She works in science industry. And our first 2 episodes, and I'm just carrying on the same podcast. First 2 episodes, one we talk about superheroes and the representation of women in science. And we also talk about how superheroes would actually have babies if they were representative of their actual names, i.e. Spider-Man. How would Spider-Man have babies if he is actually had spider qualities? So, check it out. All right. I will see you guys later. Thank you for being here. This is super awesome. Have a great Saturday or Sunday if it's Sunday there. Bye.